Hey everybody, welcome to the Growing with Fishes podcast, episode 288. I apologize for not having an intro this week. I am far, far from home, and we will. I still got to get the internet fixed out here. So, thanks everybody for watching. I'm your host Steve from Ponics. Today we have Danielle from Aquilitas. Thanks a lot for joining us.
No problem. Thanks for having me.
There's a lot of really amazing research and a key member of a very awesome aquaponic cannabis company up in Canada called Aquilitas. We're excited to have her on the show tonight. We also have Marty Waddell joining us.
Hey, what's up, everybody?
And we got Wes Engine. What's up, Wes?
Big up, Boughton. Big up, Marty. Big up, Martel. I don't know if I said that right. I'm sorry. But oh, go ahead, please.
Danielle. It's just
Danielle. Okay,
yeah.
Sorry. Thanks for having me tonight, and look forward to hearing lots and learning lots from our guests. Excited.
I'm having some internet issues out here, so I won't share the link or won't share the picture. But if you guys are looking to learn more about aquaponic cannabis and you want to take a full-length course, please check out the course that Marty and I took a lot of time to put together over at apmjclass.com if you're looking for a top-to-bottom aquaponic cannabis education session. All right, thanks a lot, Danielle, for joining us. Tell us a little about yourself and a little bit about Aquilitas. You do a ton of amazing research. I know you're going to be unveiling some really cool new stuff a little bit later here in the year. Some awesome resources for people that are interested in aquatic microbes and aquaponics and everything in that realm. Like literally everything in that realm, which is pretty amazing that you put this huge database together. And then a whole bunch of awesome research comparing aquaponics and living soil and combining those two methodologies and showing how, the difference that those aquatic microbes can really have on the soil structure and plant production and yields and all the rest of the important metrics that we're reading for cannabis production. So thanks a lot for joining us, and please tell us about yourself and what you got going on.
Sure. So my name is Danielle Maitland, and I am the Aquaponics Development Supervisor at Aqualitas Inc. We're a licensed producer that grows using koi and aquaponics water combined with a living soil. We're located in Brooklyn, Nova Scotia, so the South Shore of Nova Scotia, right on the Atlantic Ocean, and I've been working with them since 2018. I came on a couple months before we got our fish. And we are certified through Clean Green as for organic cultivation. And we try really hard to embrace sustainability kind of throughout our whole production system. We have ocean source plastics for our packaging. We use the aquaponics irrigation. We're researching how to utilize our solid wastes and process those to make plant supplement that would replace kind of more traditional fertilizers. And I do a lot of R&D. That's my primary focus of the company, and it's all about improving the quality, improving the yields, and kind of proving out the business case for aquaponics and trying to improve the profitability however we can. So I guess I can talk a little bit about our early R&D. If you mentioned some side-by-sides. So
you had an incredible presentation at the the Aquaponic Cannabis Conference and at the Aquaponics Association Conference. Both some great talks on some of the different science that you worked on, both in koi, which was an incredible talk that you did for the association. I thought it was one of the best ones of the conference. And then also on our podcast and conference as well, or at the Aquaponic Cannabis Conference, on the difference between having aquatic microbes and not. And it really was mind blowing. Some of the other metrics that you had tested that I had never gotten the ability to test. So I'd love for you to tell us about that, how you set that up and some of the results that you had, because they were quite astounding.
Yeah, so our early tests, when we first got the fish in, we only got about 50 kilograms worth of koi. They were very small because it's easier to transfer smaller fish. So we couldn't go full scale up right away, just because we didn't have enough fish to make enough nutrients. So we started out small, and one of the ways we started out was looking at your kind of just traditional living soil versus living soil paired with aquaponics. And we did a few of these trials, and the initial results were so good that we just kept on with that method of pairing the aquaponics with living soil. We've investigated other things as well along the way, but that's our primary growing method at this point. And we found increases in THC from 4 up to 20%. We saw terpenes going up, usually plus 15%. Our yield per square foot increased anywhere from 20 to 40%. And this is in the same room with the same HVAC, the same lighting, the same spacings. Like it wasn't a randomized controlled trial, which is what the later stuff we did was, but it is, it was sort of an early exploration into it. And that was enough to really motivate us to continue with this and to make that optimizing that method kind of one of our main focuses. I wish we didn't look necessarily right at the soil microbes at that time, but I hadn't heard of the soil food web at that point, so I didn't think to take samples. But it is, you're adding a living water to a living soil, and it does really, we find, improve terpenes, improve THC, improve yield, and it's quite interesting. We did a spacing trial, one of our earlier trials, where we had a different number of plants on the tables. And normally when you're doing an optimization graph, you'll get a parabola of, you see as you get a wider spacing, the plants can grow bigger, you get an increased yield, but then eventually the number of plants you have goes down to the point that your yield goes down. So it's shaped like a parabola. And what we noticed with the aquaponics water is that it shifted the parabola up and to the left, so we could get more yield with fewer plants, which is great because it means, you have better airflow, you have less cramped plants, so you're going to have fewer issues with diseases. It's less, soil you're using, it's less staffing, it's, it has a whole bunch of other benefits on top of the increased yields and the increased cannabinoids. So we, that was very early days. Our fish were still teeny tiny, but as fish grew, we were able to scale up. We're now actually, all of our flower rooms can be irrigated. We have enough fish, we have enough nutrient generation. It took 3 years, but we got there. And because we're using koi, we don't harvest our fish. They're effectively pets with jobs. We have bred them just to replace our own population as we go. And there's some adorable videos on my talk, I think, for your conference there, Steve, if anybody wants to see some baby fish. We really, we try and make their lives good. We try and do some environment enrichment, but koi are generally very relaxed fish, so that helps quite a bit. And so once they got bigger, they were making more solid waste, and so we started looking into digesting that and using that as a supplement. And that's when we got into the side-by-sides that I talked about, or the randomized controlled trials that I talked about in a bit more detail at the conference. And so the basic idea behind those, the initial trials we did, we had a few tables set up with different blocks of plants, and we compared just municipal water and living soil with a vegan commercial fertilizer compared to a fish hydrolysate, which is just, you take fish processing waste and dissolve it in phosphoric acid, effectively. And we compared that with the aerobically digested solid waste from our aquaponic system. And what we found was we got similar yields with the fish hydrolysate, which was the highest, and similar or better THC. We got some interesting grading data where the solids, or sorry, the digested products resulted in more large buds as a percentage of the total harvest. But it was really an interesting study. I know we were always aware that, you know, you see a growth spurt in after you flip the flower, and then the growth levels off. But we saw that we have, you know, a nice graph that shows us that, and there wasn't really much difference in, you know, plant height or stem diameter, but we did see changes in yields based on what fertilizers we were using. So once we confirmed that, you know, we were still getting adequate THC, we were still getting adequate cannabinoids, and we could improve our yield at similar rates to commercial fertilizers, we switched gears a bit, and we decided to look at what the optimal dosing rate is. So we had another randomized control trial. This time it was with a CBD cultivar. And we looked at, you know, high, medium, low dosing rates, or just getting water from our RAS system. And we individually harvested our plants, and we weighed out the bud yield. And we did find, you know, a dose response. We also looked at either irrigating the plants from the top or the bottom, and how that would impact how the plants grew, what height would they get to, how much yield you would get. And with this particular strain, irrigating from the bottom resulted in a higher yield, and also it resulted in more of a response. So there is a bigger difference between the different dosing levels for the plants that were irrigated to the bottom versus to the top of the system, likely because the ones that were getting irrigated from the top weren't quite as happy. So there was something else there that was holding back their yield. And so we found our nice peak of, you know, what the optimum dosing was. We did see that there is such a thing as too much of a good thing. So if we were dosing at the higher rate, we did see a decrease back down in yield. And we took that data, and we also looked at the CBD production. It was pretty stable at the very high levels of dosing. We did see a dip, likely because there was just the plants were just having issues because of the high level of supplementation. And we, so now we know, you know, at least for this cultivar, this is the dosing frequency, the dosing amount for the aquaponic solids nutrients that we can use. And so we move forward with that. And we've actually quite recently weaned ourselves off of commercial fertilizers. So our budget has gone down to maybe 5% of what it was at one point. And we're, you know, we're better utilizing the nutrients that are going into and coming out of our aquaponic system, which is pretty exciting. That was a big milestone for us to get to that point.
Have you seen increases in specific terpenes? We've seen increases in a couple of different terpenes with the aquaponics versus our controls. Is that something else that you've observed as well?
We haven't looked at that, I don't think, too specifically. I know that the cultivars we have looked at generally have a fairly stable terpene expression. So there's usually the same, you know, top three. But we didn't do the specific testing on, or I haven't crunched those numbers. I probably could, and I will now that you mention it. But we didn't look at that initially of if specific ones were increased more than others, just the total terpene content.
We've noticed transnerdal and what the hell? The other one is a fluorescein. I can't remember the name. It begins with an F. Those two in particular seem to be really boosted by aquaponics. I was just kind of curious on that. What other things can you tell us as far as the hybridization of the aquaponic water and the living soil? It seems to be, a lot of people kind of are in one camp or the other, and you're over here kind of going, hey, these work twice, even better if you combine them two, and that's something that I'm very passionate about as well. You kind of have a slightly different way to go about it than I do, but both ways work incredibly well. And I'd love for you to tell people, some of the other benefits to adding it to their typical soil regimens, because I know you kind of came to it from kind of a different approach. And Mike West says farnescine. Yes, thank you, Mike. Mike West is awesome. We got to get him back on the show too. He's one of the head people over there in the lab at Embark.
Could Danielle maybe explain her grow style?
Sure.
Yeah, so we are irrigating our living soil with aquaponics water. It's primarily a decoupled system. We do have the capacity to couple in veg, but we primarily do decoupled, and then we also will supplement with the solid waste digestate, depending on the cultivar and how hungry she is, will increase or decrease to kind of not waste anything, is the primary goal. And we do find, I like it for the sense of cannabis is still a very new plant crop at a commercial scale, especially at a regulated commercial scale. And there hasn't been a ton of research. It's getting better. There's, a lot of research facilities that are now saying, oh, we got $5 million, we got $10 million, we're opening this $50 million facility. So I'm hoping that a whole bunch of information, on optimal dosing strategies and stuff starts trickling out here soon. But there isn't a lot out there for, so it's difficult to figure out exactly what's perfect. And at the same time, every cultivar is different, and every cultivar is different depending on what your HVAC is and what your temperatures are, and if there was any stress at different life stages. So it is a very complicated plant to grow, and there's not a lot of information out there to go on that you can use for your specific cultivar and your specific grow method. So what I really like about the living soil and coupling aquaponics with the living soil is that because you have that microbiome there, if the plant needs something, it can kind of request it from the microbiome. It'll put out the exudates, and the microbiome will respond by taking what's in the soil and helping the plant access that. Whereas if you're working with hydroponics or like a sterile hydroponic system, you have to know what the plant needs when it needs it, and you need to provide it in the right form at the right time in the right amount, and without providing too much of anything else that's going to block the uptake of that one thing that it needs at that time. And so it gets very complicated and very difficult. And we have worked with cocoa, we've worked with a cocoa peat kind of mix, and we were able to grow successfully, but it is, I would say, more difficult, and it doesn't necessarily seem to have any significant benefits in terms of cannabinoids or in terms of yields, at least from our trials, from our side-by-sides. So that's one thing I really like, is you're utilizing both systems, the soil and the living soil and the living water, and they're working into each other, and the plant seems to respond really, really well to that. Hopefully that answers your question.
You mentioned bottom watering. Were you talking? Sorry.
I wanted to give a quick hello and introduce Fumidor as well. Joined us as well since we got started.
Howdy. Cheers, guys. Nice to see you. I don't mean to interrupt the discussion, but nice to see you guys. Nice to see Wes and Ms. Maitland. I didn't catch your first name. Sorry.
Danielle.
Danielle, nice to meet you, Danielle. Cheers. Back to the presentation. Cheers.
So you mentioned bottom watering, and so a couple people in the chat I know chimed in and were asking. Now, is it like a sip you use, or is it how do you administer your bottom watering?
So we were just doing drip irrigation to the bottom of the
Okay.
pots versus
Okay. Cool.
a halo at the top.
Okay. Awesome.
Never tried to do drip irrigation like that. I've always just done sips or what's the aquaponic one for it?
Ebb and flow.
Wicking beds.
Yeah.
So tell us a bit about some of the other things that you're working on there at the aquaponic stuff. I know you're quite the koi specialist as well.
Yeah, so we're always doing research. I have more of a fish background than a plant background, so I like to do a lot of trials with the fish and look at growth rates and the talk I gave at the Aquaponics Association was basically just we tracked our fish growth over the course of several months because we had kind of observed that didn't seem to matter how often we graded the koi. There's always one or two in the population who just seemed to outgrow everyone else, and they would sort of monopolize the tank and eat a disproportionately large amount of the food. And we wanted to see, how fast does this happen? How severe is the actual difference? Because it's hard to tell when you're just staring down at a tank and trying to tell how much bigger one fish is than another fish. And we know that there can be some impacts on fish welfare. If that there's really small ones who aren't getting any food, they're more prone to getting sick, and then they can get other fish sick. So we were concerned about it from a welfare point of view, but also an operational point of view of how frequently do we need to be grading these fish, how frequently do we need to be taking out the smaller ones or just taking out the larger ones. So we looked at them over the course of a few months, and we did find it didn't matter what grade, it didn't matter. They were all freshly graded into these tight little cohorts of, fish were all within 50 grams or something or so of each other. And then after 3 months, there was always 2 or 3 fish who were just way out ahead of the pack. And it happened very quickly, and it happened in every tank. So it didn't seem to be dependent on, the starting size of the fish. I think it's just something inherent about koi of one, there's always going to be 2 or 3 fish in the tank who like food way more than everyone else, or maybe they just start out a little bit bigger. That gives them a slight advantage, and then they take off. So from that,
I'm sorry.
Sorry?
Does that have to do with like the old, because a lot of freshwater fish that come from saltwater fish? Is that kind of that same mechanism you see like in tilapia and some of the cichlids, where you have a couple of them that just get kind of more hormonally dominant? Or
I don't know. I mean, they're not aggressive at all. It certainly wasn't they were chasing the other fish off the food. It was more of I'm bigger, I'm just going to go straight for the food, and everybody else is going to get knocked out of the way. So I don't know. I don't think it's a hormone thing. I think it's how food motivated are they, and if they're a little bit bigger to start, it gives them that advantage. But it definitely it's been seen in koi before. It's been documented. They call them shoot carp or Toby koi is the other term for them. And it is a kind of a known phenomenon, and it seems to be primarily related to food competition. So if you say you had a cup of food and it was tiny food, so there's a whole bunch of pellets, you'd see that spread happens slower and happen to a lesser degree than if you were giving us the same weight of food but larger pellets. So there's only so many pellets to go around, so the ones who are more aggressive get a disproportionately larger amount of food, whereas if you had smaller pellets spread out over the entire population, the rest have a better chance at getting the food. And it doesn't happen if, say, you have 10 fish and you separate them into 10 different tanks. It is a competition based thing. it was useful for us, and it was that was one of our earlier studies when the koi were still quite small, and it was feasible to take them all out of the tank and weigh every single one of them and then put them back in the tank. They're quite a bit bigger now, and I wouldn't want to do that, especially to our largest ones. They don't like being weighed very much anymore, just because taking them out, even with a koi stock, taking them out of the water, I still feel bad. So we do them more of a once a year kind of thing, of just to check up on them and see how they're growing. Grading, we'll top grade and bottom grade. So we'll just kind of look and see if there's, certain fish who have massively outgrown everybody else, they get moved up. And if there's anybody who's small and struggling, they'll get moved down to fish who are closer to their own size. And it's our growth rate has slowed down. They're about 4 years old now. So it is becoming less pronounced, but we also just had a whole bunch of babies. So it's very pronounced in the babies, and they are aggressive when they're little, quite a bit. So we had to be on top of that just to keep the smaller ones from getting bullied. And we have a mix of butterfly koi and regular finned koi. And the butterfly koi, it's like the difference of swimming in normal swimsuit versus swimming in a swimsuit with a cape on. They're slower, they get to the food less, they're less competitive. So pretty much all of our butterfly koi in this cohort are quite a bit smaller than the regular ones. So we're kind of trying to help them out a bit by keeping the big guys, getting them moved out as they get big, just so the littler ones can have a shot. But I do enjoy that side of it. We're also looking at, and I'm doing a PhD through Dalhousie University, and we're looking at designing fish feeds specifically for aquaponics. And we should be wrapping up the last of those trials here in a month or two. And it's something that, we're really excited about. There's been a lot of talk in the aquaponics literature for decades at this point of, somebody should design an aquaponics fish feed. That is the next step. That would be a great area of research, but there hasn't been a lot of research published about it. I know you mentioned, Steve, that you'd heard of some people trying it out.
Yeah, so Aquaponic Source actually was working with ABC Organics for a little while before the owner of that company passed away. And they were trying to work on it for a bit, and then it kind of became like a money pit, and they kind of backed off. And then that was back when Sylvia was still there. And then there was Optimal has a food that I think is economically less viable than currently. I mean, if you go and spend the minerals to supplement the same thing, you're going to spend 90% or anyways 40% less on your monthly fish feed. And then you have some others that have dabbled with it. I know Star Milling was playing with it, and what's the other one? There's one other one that was out there was trying to do an aquaponic feed. So especially for more performance plants, not so much lettuce. But no one's, people kind of did it for a while, didn't really get the results that justified enough continued funding, and gave up on it. So
yeah, and it's and I mean, that might be what happens with us. We're seeing some good initial results, and controlling phosphorus output and sodium output, which are good. But I am very much hoping to publish it, because there hasn't been a lot of stuff that's been published. And even if I find doesn't do anything, don't waste your time, at least there's an official publication saying doesn't do anything, don't waste your time, instead of the current state, which is everybody saying that somebody should do this, and then it hasn't been published yet. So that's kind of what I'm hoping to finish after 4 years of paying tuition. But we're getting some good initial results. And one thing, even if we get nothing else out of it, our koi absolutely love our fish feed. They are not fond of some of the commercial controls, but as soon as the stuff we formulated hits the water, they're going straight for it. So if nothing else, it's a good treat. But I'm fairly happy with how it's performing so far. We've been focusing on fish only trials, and then we're going to move into to plant trials at different stages of the cannabis, and we might do a little bit of vegetables as well, just to see if it has an impact on that. So that's really exciting, and I'm really happy that we're moving forward with that so far. We currently make quite a bit more solid waste than we have space to process, and because we've had such good results with the process stuff so far, we're looking into some methods of scaling that up, which will be very exciting going forward. I think based on our numbers so far, we'll probably be able to make more than we need on site. So either making it into a product, or using it for an expansion, something like that is that's on the table. And I'm excited about doing that research and figuring out a way to commercialize it, because it is applicable to not just aquaponics, but to any kind of freshwater aquaculture facility can use similar processes. And it speaks. Sorry, go ahead.
I was gonna say that I know the main chemical challenges you run into are the iron oxidation, the potassium being a heart problem issue for the fish, and the manganese also, dropping out from oxidation. I often wondered if you could do fulvic or humic chelated versions of those and have them pass through the fish and remain in a plant usable form, but I don't know of anyone that's had a chance to test that for that application. I do know 2 people making pretty good quality humic acid chelated iron and some other stuff now. It's something I want to try, and I'm real curious about. Have you worked with any of that type of stuff, or is it something you can't talk about or anything?
We did some initial trials just looking at combining some iron, some commercial iron based organic fertilizers with our digestate, and the iron grabbed the phosphorus and fell out of the water pretty quick, which is probably a chelation issue. So I could see humic or fulvic helping with that. But that is one thing you see of, if you have a high phosphorus, you don't have much iron, and then if you add, it's actually a method for wastewater treatment plants to pull phosphorus out of the water before they discharge it, is they use an iron product that will bind to the phosphorus and follow to solution. So it is definitely a well-known issue with your more available forms of phosphorus, that they're very friendly with iron, with calcium as well, but definitely with iron.
Funny, they use phosban reactors for reef tanks for the exact same purpose of stripping the phosphorus out of the waters.
Yeah.
It's really interesting. I never thought about it in that plant context.
No, there's a lot about the digestion processes that are very similar to things that you'll see in human wastewater treatment. My background is in chemical engineering, and I've done a few courses and projects on human municipal wastewater treatment. And actually the poster behind me is all the different types of bugs you'll get in your activated sludge process. And it overlaps exactly. If you take a happy aerobic digestate and look at it under a microscope, you will see pretty much everything on that list, and you'll see the progression of the populations happen in the same way that you do see with an activated sludge process. And it's kind of based on the amount of energy that's available at the time of, you'll see first you'll see your fast moving, the bugs that have to do a lot of work to eat. So your ciliates that bounce around the microscope slide really fast and have a million legs that are pushing them all over the place. Those will start first, and then as the amount of food goes down, they sort of start to drop off, and your things that do less work to eat will show up. So your filter feeders, I'm blanking on the name. But you know, they kind of sit their feet down and then they just kind of pull everything into their mouths. You'll see those come up, the populations of those come up, and then you'll get your larger predators, so your worms and your nematodes, coming in and sort of cleaning things up. And then as that progression happens, more and more of the nutrients are getting released into the liquid form of your digestate, because, you know, as you go up the food chain, they become less efficient. They don't need, you know, as many of the nutrients to create their own bodies, so they are releasing the excess in generally a plant available form. So sorry for that rant, but it's a really interesting thing, and it's something that it's really fun to just look at under a microscope, because you can see it changing in real time. And you know, even if you have just a decent light microscope, you don't need something that can see and count bacteria, or you don't need to do any staining. It's just a matter of you know, putting a couple drops on a slide and turning the light on.
I had a question for you, just because your data very much contradicts something I heard the other day. So I was listening to a podcast, I think it was on FCP, with Nick from Rooted Leaf and the gentleman from BioAg, and I believe Glenn and Rust popped on there too. And they were talking about how the nitrate pathway for conversion was actually directly competing with terpene expression and the resources for the nitrogen used for terpenes and cannabinoid production, and how those different pathways worked. But with your results, you're actually seeing the opposite. You're actually seeing improved numbers with nitrates being the primary nitrogen source versus from the fish waste versus some others. So it kind of made me think like there's got to be a lot more into that mechanism than we kind of currently understand with the plant science, and how, you know, there's a lot more science that we need to learn now that we just have no idea on, what these other forms of microbes or other plant processes that were currently not understood. So I didn't know if you had any more thoughts on the nitrogen stuff, because typically people always talk about nitrates being a little bit harder on the plant, and so, you know, some kind of other negative things around nitrates. And again, I myself think that it really is the better way to go for production. You do as well. I wondered if you could speak to that a little bit.
I haven't seen the research that you just mentioned. I do know that I have seen some stuff come out about, you know, if you increasing nitrogen will have, you know, a decreasing impact on THC, and specifically in the form of nitrate. But I remember looking at that, and the concentrations of the nitrate that we're using weren't at that height, weren't at that level. So that's why potentially could be why we weren't seeing a negative impact. We do have a little bit of ammonia in our water as well, so it's not all nitrate, but it is predominantly nitrate. And we haven't, I mean, our results have been good in there. I know that the living soil does have you know, chicken manure in it, which is quite high in ammonia. So perhaps that's countering it a little bit of, you know, we're giving them both. But I haven't seen anything side by side. There is so much research that could be done that's interesting, and so much that, you know, it just needs a proper, it's difficult to control, and especially with living soil. Or with soil in general, it can be so heterogeneous. It is a difficult thing to control, and the plants themselves can be quite difficult, depending on you know, how the clones are taken, and how you know, even your canopy is, how even your plant sizes are when you start. We had some issues with our controlled randomized controlled block designs, where we'd have, you know, 2 or 3 plants throughout the grow that just were stunted. Not necessarily sure why, but that does impact how you have to do your statistical analysis, and you have to look for outliers and whatnot, and that can be, it makes everything a lot more complicated. And when you have a plant like cannabis that is, you know, I don't want to say difficult to grow, but so heterogeneous just between different cultivars, it is hard to get, you know, you can say for this ghost train haze, or for this Charlotte's Angel or for this, for my cultivar, nitrates are bad. But if somebody else could have a different cultivar with the same name, that maybe that's its real name, maybe that's its real, parentage, or maybe they just decided to call it that because, or somebody told them that's what they got, that really likes nitrates, or, really likes bottom watering, or really likes top watering, or, responds to drought stress with an increase in THC, or responds to drought stress with complete nutrient lockout and, no THC production. It's such a varied, there's so much variation within the plant and between plants, and between cultivars, that it's a really interesting area of research, but it's going to be difficult to see how translatable it is, when you have everything is so specific to what you're growing.
It's a great point. As we, people often ask me, like, is there anything you can't grow in aquaponics? And it's like, yeah, well, like 4 or 5% of all phenotypes are just going to hate whatever it is that you're doing. So if you're growing aquaponics, it wants to be in soil. If you're doing soil, it wants hydro. If you're doing hydro, it wants to be in soil. Like, you know what I mean? So that you do have a couple, 5% that are just, pouty plants. You know what I mean? And you just can't do anything about it.
Yeah. And we do all of our, or not all, we do a lot of our own pheno hunting in-house, using our process, using our procedures, so we know how. So we're looking at how it's going to respond in our system. So we know, this cultivar, we got great terpenes, great THC, great yield, really good structure using our soil and our aquaponics water. So when we go to translate that into a commercial setting, we don't get any surprises.
Did you do any comparisons on trichome yield, trichome diameter, between different ones?
I would love to, but I don't think I have the equipment for that. You probably need a.
Oh yeah, just need a set of bubble bags. In fact, we'll hit up Marcus. We'll see what we can do for you.
Okay.
Yeah, we'll hit
I'm
up
always
bubble
looking.
bags.
I'm always looking for an extra thing I can test. So.
I know. Mike Westover in Embark is a big fan of aquaponic cannabis. He said that it's the best weed he's smoked in all of Canada, or anywhere really. Has been aquaponic weed from Canada. So a big fan. I'm sure we can find a way to. If not, I'll send you some.
Great. Yeah. It's always good to come up with, and there's not. I wish there were more metrics of things, because right now it's basically, you can get your THC, you can get your cannabinoids, you can get your terpenes, but in terms of things that are easy or don't cost $150 to test, I guess.
Sure. Well,
is the way,
the nice thing, if you bubble washed your stuff and check the size, you could actually see what would be better at yielding per hash. So it'd be very relevant, and it's the cost of water and ice. It'd be a very good affordable test to try. I'm certainly curious to see if it's something that I've never done with direct with any of our soil and aquaponic comparisons, and it's something I've always wanted to do, and then I always kind of forget after the fact, because you're always worried about all the 10,000 other things you're trying to record for whatever study or whatever else that you're working on. So it's easily lost. One other thing I wanted to mention real quick about bubble bags: if you're traveling around the world and you want to meet with some heady weed people out and around the world, and you want to trade them to get good seeds, bubble bags are one of the best things that you can bring through an airport that you're not going to get harassed about, and you can trade, and it has trade value that is on par with bringing seeds from the States, right? So if you're traveling to Africa or the Caribbean or something, and you don't want to pollute the population's gene pool, you can absolutely trade bubble bags, and it has very good trade value for the people that actually understand the value of them. They'll go blue, you got that here in the middle of random faraway place. How did you do that? Anyways, so I've had a really good luck with that, and you can get bubble different sets of bags for fairly affordable prices these days. So definitely an option if you do want to be a more conscientious seed collector. Do you want to tell us a little about that incredible database that I know you kind of touched on it a little bit earlier, but that incredible database that you're working on that you'll have out for us soon, or?
Yeah, sure. So as part of my PhD, I was working on a literature review, as you do. And I started, I'm a big fan of Excel, so I figured, okay, I'll just, as I go through these, I'll put everything into Excel. So I started out fairly small of, I'll put in the title and then, the paper and the authors, and it basically turned into this absolutely massive sheet that has all the parameters from the aquaponics literature. What plant were they growing, what spacing were they using, what fish were they using, how big were the fish when they started, what density were they growing at, what size of fish tanks were they using, and it just kept getting bigger and bigger and bigger. And I went through basically all the literature from mid 2021 back to the start, which the first articles were in 1977, 1978. So I have all these parameters scored for each paper for about 300 papers. And so I'm going through now and making sure checking for typos and spelling mistakes and filling in some things that I decided to add in halfway through, just going back through the first papers I looked at. And it has the potential, I think, to be really useful for people who are either, looking at doing commercial aquaponics or looking at doing aquaponics research, or they're looking at publishing their aquaponics research and they want to be able to compare it to what people have done before. So say you're growing tilapia and you want to know all the papers that have looked at tilapia. You can search the spreadsheet, sort it by just show me the tilapia papers that we're growing using, lettuce and fish that were over 50 grams. And you can sort, and then that'll give you a list of references that you can look up and look through and say, how close is it to what I'm doing? Or how, how did they set up their experiments? Or, what did they find? Was it, or what were they looking at? Were they comparing, different pHs, or were they looking at different stocking densities, or, different, were like, media-based versus NFT versus deep water culture, that kind of thing. I'm hoping it'll be a really good resource for people who are interested in aquaponics, or who are researching or looking to do commercial aquaponics, just to speed up their literature search, so they can kind of at a glance tell what is most relevant to them. And so the plan is to do to pretty it up and make it a little bit more user friendly than it currently is. And I'm hoping to post it just out freely on the Aquaponics Association website by the end of the summer, hopefully. But I wouldn't recommend it. It was a lot of work.
I'm a huge spreadsheet nerd. I've been working on the Open Nutrient project to try and aggregate all the different mulch and liquid fermentation and compost tea data into one central database. So I feel your pain.
Yeah, it's when you say, oh, I'm just going to get everything, it just tends to cascade on you. But what's your favorite from doing that? What's your favorite compost tea?
Compost teas, I'll be honest with you, I've kind of moved away from them, other than a couple of scenarios. I'm much more, I think, if you're looking for the same kind of benefits of compost tea, I really feel like liquid IMO is a much better product, and a much better thing for your living soil and organic, sustainable soil production. I've seen IMO do all kinds of crazy things and cure different diseases much faster than any commercially available product, and cure things that allegedly you can't cure once they hit a certain point of infection. And after that, it's like, well, all right then. This is simply fixing the food web seems to do a whole lot more than we realize. So it also allows you to use less nutrients and everything else. I know out in Oklahoma we have a real bad problem with disease called septoria, But if you spray your fields ahead of time before you even put the seeds in the ground or clones in the field with liquid IMO, you can very, very easily, completely prevent the septoria outbreak entirely, because the plants have that biodiversity in the root system that helps their immune system, defend itself. So it just helps boost the plants in a way that is just much, much better. And then even in some of the controls that I've seen out there in terms of comparisons, the side-by-sides with and without, you get higher terpene levels. Again, for the same reason that aquaponics gets higher terpene levels and living soil gets higher terpene levels, that you have that microbial biodiversity that's significantly higher than, hydro or, running GH or any of the other, generic chemical nutrient solutions that don't have any microbial component.
Yeah.
So what are some of the other things that you've, oh, go ahead, I'm sorry.
Oh, no. Cool. Go ahead with your question.
I was just gonna say, what are some of the other things that you've noticed with aquaponics as far as differences with the aquaponic plants versus your controls?
Well, actually, we didn't realize this till much later. So you're familiar with the Canadian model of when you go to batch release commercial product, you have to test absolutely everything.
Yep.
And one of those absolutely everything's is microbials. So we did our side-by-sides, and because we grew them in different ways, we had to batch release them as different products, basically. Well, they're the same product, but different batches. And so we got the micro testing done, and we did it. So on the 2 tests we did, and 2 test isn't science, but it's an anecdote or 2 anecdotes. We did find that the microbial levels on the final product, grown in the same room on the same HVAC, taken harvested at the same time in the same way, taken the same dry room, the ones that received aquaponics water had lower microbial levels on the final product than the ones grown right beside them that were just living soil and municipal water, which we thought was really, really interesting. They were still, the levels were all still fine for releasing them, but the aquaponics came lower both times, which is a super interesting thing that I'd like to figure out exactly what the mechanism is for that. But it could be any number of things. It could just be, the plant is overall healthier, so it has, a better resistance to microbials, or maybe it dries better or faster, or it could be any number of things. But the result that we came up with that we didn't actually notice until quite a bit later, because we weren't looking for it, was that the aquaponics product had lower microbial levels on the final dried product. I don't know if you've seen anything like that, but we were pretty excited about it.
Yeah, the thing I've noticed the most with it is the resistance to powdery mildew seems to be significantly higher than the soil plants that we, and there's definitely a microbe that's triggering that, that's boosting that mold resistance of the plants that is pretty easily seeable. I know we actually did some tests where we took an infected plant and tried to rub it on some of the plants to try and infect them, especially when we were testing silica, because we were trying to see, how it was a random observation that the plants with higher silica levels weren't getting it either. And we were trying to kind of force it and kind of, see how much we could push it. And it was kind of crazy to see stuff just slough it off and not even care, and still is an interesting thing that we definitely observed on that. And then some of the other stuff was just a much stronger smell in veg, a much more potent of those non-sessile trichomes, and some of those early trichomes that form on the plant, especially doing stem rubs. You definitely get a much more fragrant preview out of the aquaponic plants, I've noticed, in the soils. Again, minor observation, but it means that they're producing more,
Yeah.
Even in that early stage. The other big thing that I've seen sometimes is in certain strains, as much as 100% increase in CBD. We've had some strains go from 2 to 6% and things like that. Which is, again, I think directly attributed to some type of aquatic microbe, changing the expression of that. So, and again, this is all stuff that just needs to be fleshed out. A lot of hard questions there to try and figure out how you'd even quantify a test for that.
Yeah.
To try and determine what it is. We did have some questions from chat. Jim Owen asks, what are your different things that you could add to soil to increase terps and or THC in cannabis?
Well, aquaponics water, for one. If you're looking at more of a home grower situation, and this is, you can, if you have a fish tank, that's not a saltwater tank, you can use water if you're doing a water change, you can use that as your irrigation water. That's something that I do at home. It makes my goldfish happy because they get water changes a little bit more frequently because I want to use their water for growing things. I know, I can't remember, and Steve, you might have seen this, there were some, I feel like there's some research that came out about worm compost, or might have been compost teas and THC expression, but I can't remember that well enough off the top of my head to speak to it.
I know there was one with, was it chamomile? I think it was being interplanted with the plants, increasing THC expression. I think that's the only one I can remember seeing off the top of my head. But yeah, I haven't seen one with the worm castings. Man, it's a shame that Coot's not here. Coot would know that for sure. In 3 seconds, he could rattle off the paper on the worms. But he's not here, unfortunately. But that's the only one that I guess comes to mind off the top. There isn't a lot of studies on individual triggers around THC, and which is makes it kind of hard to try and suss it out, other than just making sure your manganese is, above threshold. That is one thing that'll definitely inhibit, an otherwise healthy plant in terms of expressing THC, is if your manganese is either in an unchelated form and not bioavailable, or too low. You have to have it above a minimum threshold to trigger that, full THC expression. And if it's not, that was one of the reasons why when we were in Zimbabwe, we specifically chose a previous tobacco field that had been sitting fallow for 30 or 40 years, was because it had a ton of manganese. They dump a lot of manganese in the soil when they grow tobacco, because it's another resinous plant. So, we didn't have to really do anything other than just add the microbes to the soil, and the fertilizer was already there. So it was kind of nice.
Yeah, and one thing you can do is just, if you have one cultivar, you can do different experiments with that one, because we've seen that the same, genetically identical plant, if you grow it one way, you'll get really high yield and 14% THC. And if you grow it a slightly different way, you'll get slightly less yield and 25% THC. And it is, it wasn't same soil, same everything else, just the, irrigation method, or, how much drought stress, or how much, there's a whole bunch of different factors. So if you have, one plant, it is worthwhile to work with that plant and try and see if you can optimize it in different ways. And it might, we have another cultivar that we did the exact same thing with, and it was 24% the first time, and it was 24% the second time. It had almost no impact. So, it is specific to the plants you're trying to grow. And it would be nice to see somebody do, same room with 50 different, cultivars, and just to look at what impact do you get doing these exact same things that, we found before that have an effect, just as a fun, probably very expensive research project.
Oh yeah. That'd be a lot of, unless you had an HPLC, that would not be fun.
No. And if you had an HPLC, it would be a different type of not fun, because you'd be on that HPLC for several weeks.
Yep. Oh, first world problems, right?
Yeah.
Anything else you wanted to talk to us about as far as your stuff? I know it's getting a little bit late there. I didn't want to keep you too much past an hour or
Yeah,
so. I know it's quite late on your time zone there.
It is getting fairly late. No, I mean, I'm always happy to answer questions. If there's any more from chat, throw them at me. But
we had one question. Some people use, okay, what do some people use instead of molecular sieves?
Oh, that's not a me question.
Yeah, I'm not sure on that one. I'm not quite sure what you're referring to there, Russell. Maybe you want to clarify your question. We had another person ask, do you reuse the water from your runoff after it goes through the soil?
We don't do too much runoff. So we're most of it's going into the soil, and then the plant transpires it up into our HVAC. We are looking at it's been a pet project of mine to do water recapture from our HVACs and reuse that, but that's still ongoing. We're making some progress, but it is something that, you know, it's a good way of saving water, but it's effectively, you know, distilled water when it comes out of there. But you do have to be worried about some diseases like to live in those types of equipment. So you gotta, you know, get a good testing and sterilization process and in stream before you can reuse it.
But another question, do you have to do anything different with your pest control because of the way that you're growing?
Well, because we are certified through Clean Green, there's a bunch of, you know, pesticides that aren't approved for organic cultivation that we can't use because we are in Canada, and Health Canada controls your cultivation. There's a very small list of things you can use according to Health Canada. So we're restrained there. We have a pretty strong IPM program. We use a lot of beneficial bugs in our grow rooms as preventatives and as treatments, just to keep anything and everything under control. We are very careful about, you know, if we are doing any kind of research of what can make it back to the fish. Just because you do have to be concerned about that, the fish, as well as because we run a recirculating aquaculture system, we're dependent on a biofilter to process the ammonia that the fish release into nitrate, and that biofilter can be quite sensitive to all sorts of things. So you have to be very careful of what hits your biofilter as well as what hits your fish. You know, if you have fish, do not have yucca extract anywhere near you. Is always a good rule of thumb.
Oh yeah, I get at least 2 to 3 calls a year on that one. That's always like, well, buy some more fish. Yep.
Yeah, no, I've heard some horror stories, so I don't.
Carbon does a pretty decent job of pulling it out after a little while. So.
Yeah, one of our soil guys came and said, "Oh, I heard of this. I got this new product. It's supposed to be great wetting agent. It's organic. You know, it should be fine with your certifier." And I was like, "Is it yucca?" He's like, "Oh my God, yes, you've heard of it." And I was like, "No, thank you. I like my fish."
Oh yeah,
I'm very fond of my fish.
I always love explaining to people how the Native Americans on the west coast of the United States used to collect yucca roots, squeeze the juice out, concentrate it into clay vessels, evaporate it down, and pour it into rivers during the salmon run and kill off a section of the river. And the rest of the village would be waiting about half a mile downriver to collect all the fish that float to the surface so they could smoke them and eat them. And it's like, yeah, that's how poisonous it is. Like a few drops will kill thousands of gallons. Like people really underestimate it.
Yeah, no, it's one of those things that scares me, for sure. So we're pretty careful about that, and we're.
What's interesting is you know beetle nuts, like the kind that like people in Thailand and Indonesia like get high on, it rots their teeth out and stuff. They actually take an extract of the beetle nut husks and they use it for killing planarian and worms and nematodes. But that is a wetting agent as well and isn't fish lethal. It actually isn't shrimp lethal either, or coral lethal if it's dosed properly. So it's a very gentle, it is one of the weird things that's a suffocant that's from the same kind of chemical base from a nut or a root that is, you know, safe and can be used to treat, you know, parasites in that way. I use it a lot with those little ornamental shrimps, the cherry shrimps and things like that.
Yeah.
When they have the planaria that are killing them. Someone asks, will any wetting agent do that or just yucca? Most wetting agents are incompatible with fish keeping in general. Yep, unfortunately. It just is part of how the chemistry works, unfortunately. It allows them to, kills them through a couple of different means, but yeah, without getting into the science on that. Any other things that you'd like to share with people about aquaponics or living soil? They're combining the two, or people that are interested in learning more about this.
Well, I gave a pretty good talk at the cannabis conference that Steve put on there, and I go into a little bit more detail in some of our trials. So if you want to see that, there's also a few talks at the Aquaponics Association that I gave that went a little bit more into koi. I can, if you're in Canada and you want to bulk purchase some baby koi, we can probably help you out if you want to reach out through our website. I am realizing that I know absolutely nothing about how colors are inherited with koi, because we had two parents that were, one was kind of a platinum mirror koi, and one was white with very bright orange fins, and we have blue. We have koi that are blue with blue scales, which is not something I was expecting. So we've got some really interesting looking fish. I'm very fond of the babies, and we're looking to maybe sell some, just because you don't want to have 2,000 of the genetic siblings holding up your aquaponics operation. That's not a lot of genetic diversity. So but we have
limited drop aquachronic koi.
Exactly. So as far as I know, we're the only people in Nova Scotia who breed koi. So there's that. There's there's a lot of good talks. I know our social media is probably going to be pushing more pictures of the babies at them in the coming months. So if you like that, feel free to follow our socials. If you want to try our products, we're available in most places in Canada, most provincial distributors. We got dried flower, oils, vapes, gummies, rosins, all sorts of stuff available. And all sorts of different cultivars with different terpene profiles and cannabinoid profiles for all your tastes. In terms of the R&D, hopefully we'll be getting some more interesting stuff on that as we move forward. And I'm looking to start some more blog posts and stuff like that with pictures and kind of brief descriptions of studies and results, and hopefully add to the kind of the non-scientific literature on the subject. And I'm always happy to come on things like this and talk about our fish and our process and some of our R&D. So thanks for having me. It's been fun, even though it is pretty late in the evening. Hopefully I'm not getting to the point where I'm babbling yet.
No, you're fine. I really appreciate you taking the time. We're definitely welcome on here anytime, especially when you want to tell us about some of your newest research. We're always happy to hear about all the awesome work you're doing. Definitely hope to get you on for this year's Aquaponic Cannabis Conference. We're definitely going to be doing another one this year. I usually kind of wait till after 420 and planting season's done to start organizing that. So we will be taking proposals for presentations this year, and I definitely want to get you on there again because really learned a lot with your presentation last year. So thank you so much for coming on the show, and really appreciate your time.
Thanks for having me. It was fun.
Thanks a lot. And you're on one of those funky half time zones, right? 30-minute zones? Or is that the other part of Canada?
That's Newfoundland. So
Newfoundland, that's a little bit farther east, right?
Yeah, it's currently 12:48 a.m. for me.
Okay. Well, thank you so much for being awesome and sticking with us.
No problem. If I come on again, maybe just push it back a few hours.
Oh yeah. We'll pick it up an hour for you next time. I apologize.
No
It
problem.
ready to work. It probably wouldn't have helped today because we had all the technical issues, but that's okay. Thank you so much.
No problem. Have a good night, everybody.
Take care.
Bye.
Well, that was wonderful. Always love having Danielle on the show. Always enjoy having her talk. If you haven't already, please go check out her talk at last year's Aquaponic Cannabis Conference. It is a really, really wonderful and very informative presentation. How you doing, Wes Engine? How you doing? What's new with you?
Man, I am high as giraffe dick. I've been smoking fucking Lime River Rose all night. I am fucking stoned. But that was an awesome talk. She was a great guest. She was so knowledgeable, so smart. But yeah,
Oh
no,
yeah,
that was
she
awesome.
and what's cool is a lot of people listen to the show. I mean, we kind of mainly aim the show at living soil and aquaponics, and she kind of does a whole different way of hybridizing those separate from the dual root zone stuff, which we've all talked about so much, and it kind of has its own benefits and things like that. So it's kind of unique and neat to hear about, and kind of just seeing how again those microbial diverse communities in the soil really are the best way to go, no matter how you go about achieving them. What's new with you, Fumi? What's going on in your garden? I guess he stepped away for a moment.
Yeah.
It's okay.
I just harvested that Oaxacan Taya is growing, and a bunch of other stuff. Some black garlic, Jordan of the Isle. I got a bunch of other stuff flipping and going into flower. A bunch of skunk stuff going into flower. Yeah, no, I got the garden's pumping right now. So
Nice.
pumping right along. Yeah.
We did. I'm working on a new project right now. We'll have a bunch more footage on that soon. You got a little bit of a preview on Instagram.
I
But
seen that. Yeah.
we did a quick trial on. We just had a bunch of different seeding mediums, and we were supposed to get an order on Tuesday, and it's probably going to come next Tuesday, and I'm out of plugs. So uh, we just used what we had laying around, and we did a bunch of we did some in the Oasis flats, and we did like one or two Oasis flats, and then we did rock wool, and then we did rapid rooter, and I got a little video on that. That'll be going up on YouTube here in the next day or two, when I have time to sit and think for 5 minutes. And then although tomorrow is supposed to be a pretty big tornado outbreak, so we'll see how that goes. And then we'll just have a lot more plant footage and stuff, especially as construction stuff goes along. We have a big outdoor living soil project I'm going to be showing you guys. It's going to be a little bit bigger than probably most of the other stuff on YouTube right now, size wise. So I'll leave that as a tease for now, but it's going to be pretty big, so that'll be fun. And some other really cool stuff down that space. So other than that, yeah, just have some other things in the works. We have Tommy Chong coming on the show May 12th. Super looking forward to that. We should be going back to Monday and Thursday shows here in the next week or two. Might be another week before we're doing it back to 2 shows a week, just because I'm kind of living in 2 places at once, working at this spot a little bit farther away. So depending on which spot I'm at, it's a little easier for me to have a connection. Like right now, my internet connection is not the best here, but it'll be better when we get the new service here pretty soon. But when I'm up at the other, my normal house where I live, we'll be able to do episodes on the normal schedule. So bear with me here through the spring, and we'll be back to normal here before too long. Other than that, you back there, Fumi? Not going, but other than that, I just gotta, hopefully I'll be able to pop a lot of different seeds. I have a ton of stuff in my collection. It's starting to get a little older. I want to start popping at least some of it and seeing what's going on. So trying to pop a bunch of that. Doing a little pheno hunting and some other cool stuff. Gonna be trial running some different autos, so that'll be fun. I got some Lion's Claw from Pride of the Lion Seeds. He was on the show a month or two ago. I don't remember exactly.
I remember I was here when he was there.
Yep, and then I got some auto seeds from the gentleman out in Maine. I'm gonna be running some of those and some other stuff. So excited to run some of that. I'm not a huge auto fan, but excited to run theirs, and I'm looking for some consistent stuff. If I can find stuff that's consistent in size, then I can run it commercially. But it's it's been kind of a crapshoot with all the stuff I've tried to date. So I'm excited to run those. We actually have a bunch of Lion's Claw running as well over at Vertica. So we'll have two different, Bane running it over there, and I'm running it over here. So we'll have kind of a broad, two different personalities on growing. So that'll be fun. What else? That's about it for now. We'll be doing a monthly grow club with Jordan River here before too long. That'll be fun. And trying to think what else. Look for announcements on that. And then other than that, I'm just super stoked to have Tommy Chong on the show. It's definitely someone who I've been listening to religiously every Christmas. The Cheech and Chong a Christmas album, and my dad would put that on. That's like part of Christmas to me, and it'll be really amazing to have him on. It's funny enough, I was with a friend of mine. We dropped some stuff off at Tommy's house one time when he wasn't home. But his wife is amazingly pleasant person. She's fucking awesome and super sweet. And excited to have Tommy on. I don't know, that's kind of a super cool thing for me, and excited for that. And
Absolutely. Absolutely.
Yeah, and that just cool. Been busy working, just kind of juggling 3 projects right now. Everything's kind of going on all cylinders again. Kind of nice to be back with more work than time to think. So, and then I'll be honest with you, it's nice to be back in the grows every day. For a while I was doing a lot of design work and consulting work, doing other stuff, and not a lot of what I like to call plant therapy. It's nice to spend all day with plants again.
Yeah, man. It's
certainly made me a
nice to be in the forest, man.
Oh yeah, and then plus, like at the one of the 2 spots, and we're kind of bouncing between the nursery and the main farm that we're finishing building out right now up here, and we have like hundreds and hundreds and hundreds of acres, and with the 2 wolf dogs, they can just run and chase the deer and go have fun and be dogs. So that's really, really nice, and just
that's awesome.
Been kind of a chill week all around. So. Tomorrow we have a huge tornado outbreak, and then I think Monday or Tuesday we have another big tornado outbreak. So enjoy the calm while we can. Alrighty, Fumi, are you back? If not, you're gonna have to come back for the end of the show. trying to get your seed site up here. If he doesn't come back, please go check out fumidoro.com. He has great seeds, including Lime River Reserve. In fact, why don't you tell us a bit about that, Wes?
Yeah, Lime River Rose. I've grown that. That's absolutely beautiful plant. The incredible lime phenos are just in there. There's everything ranging from lime candy to lime pop. I found an amazingly interesting and unique rose pheno in there. It came in with just a lime on the front and then a potpourri on the back, but then it lost all the lime, and it smells like a bouquet of flowers when you smell it. It's like, I've never really smelled weed like this. It's very perfumey, and it is super stony and just relaxing when you do smoke it. It's really good weed, really awesome weed. So if you're looking to try something new, just want some beans to hoard, check out Fumidor seeds. Yeah, man. chronictable.com, a little easier to find it that way. And yeah, check out the West Engine Swindle over
there. How do people find you and all your cool stuff?
Oh, me, I have one of those Instagram things, West Engine, WSINGIN. Yeah, man. And yeah, about it. And I hang out Tuesdays, Wednesdays, and Saturdays on the Chronic Table over at Fumidor Show. So come over, stop by, hang out.
Yeah, I'm usually over there too. So I've been a little bit busy this week, but normally I'm over there at least most of the time. So always fun hanging out over there as well.
Yeah, man.
And you can find Fumi at Fumidoro or Fumidor_Chibador or Portland Tasting Society. Portland Cannabis Tasting Society, Portland Taste. I forget. I'm sorry. See, this is what happens when he's not here. It's okay. It's fine. It's fun to give him shit though. What else? You can find Danielle at Aquilatos Inc. We'll have her info in the chat there, in the description. And you can also find her Reef Organic and Aquilatos up in Canada. They're kind of the same company ish. And then one's different parts of the market. Long story. And then you can find us. What else is there? Who else was on the show? Marty was on APMeds. You can find him at APMeds on Instagram. And you can find me, Ponics, SoundCloud, YouTube, iTunes, all the things. I'm working on some recorded outros and some recorded intros, so I don't have to say that and have it nice and professional and all that. So I am working on it. So as soon as I get back home and a little more time in my normal studio, we'll get that going. And yeah, if there's any guests that you guys want, I do have quite a bit of a queue currently, but if you guys have any guests that you want to see on the show, feel free to let me know. And we'll see you guys again next week, hopefully Monday. If not, I'll see you guys on Thursday. Yeah, thanks a lot, everybody. We will catch you guys again next time.